Hello everyone, and welcome to our channel, MooTechie. It's great to have you here. Today we're going on a fascinating journey into the molecular world to explore the incredible molecule known as RNA. Let's dive right in. So, what will we be covering today? Here's our roadmap. First, we'll start with the definition of RNA. Then we'll examine its general structure and components. After that, we'll look at its classification, focusing on mRNA, tRNA, and rRNA. We'll explore its functions in gene expression. And finally, we'll wrap everything up with a summary and key takeaways. Let's get to the heart of the matter. What exactly is RNA? The core definition is this. RNA is a polymer of ribonucleotides. Now, let's look at the molecular structure image on this slide. It shows a simplified but clear diagram of a single-stranded RNA polymer. It's depicted as a twisting line with nodes, and these nodes are labeled with the nitrogenous bases. We can see the bases adenine, guanine, cytosine, and uracil represented. So, to reiterate, RNA is typically single-stranded, and it uses the base uracil, that's U, instead of thymine. Its sugar component is ribose, which has a hydroxyl group on its 2' carbon, and the backbone is held together by phosphodiester bonds. And most importantly, RNA plays a central role in gene expression and regulation. Now, to really understand RNA, it helps to see how it differs from its famous cousin, DNA. This slide presents a clear side-by-side comparison. On the left, we have RNA, or ribonucleic acid. It is single-stranded. Its sugar is ribose with that 2' OH group. Its bases are adenine, guanine, cytosine, and uracil. And its nature is dynamic and transient. It's often made, used, and then broken down. On the right, we have DNA, deoxyribonucleic acid. It forms the famous double helix. Its sugar is deoxyribose, missing that oxygen on the 2' carbon. Its bases are adenine, guanine, cytosine, and thymine, and it is stable and meant for long-term storage of genetic information. Let us now look at the general structure of RNA in a bit more detail. The fundamental unit is a nucleotide, which consists of a nitrogenous base plus a ribose sugar plus a phosphate group. This slide has a helpful diagram. It shows a chain of 2 nucleotides linked together. We can see the direction of the chain is labeled 5' to 3', which is the direction it is built. The bonds connecting them are called phosphodiester bonds. One nucleotide is highlighted showing its parts, a phosphate group, the ribose sugar, and a base which is here adenine. The next nucleotide in the chain has the base uracil. The diagram also reminds us that base pairing, specifically A with U and G with C, is what enables the RNA strand to fold into complex shapes, and that little 2' OH group on the ribose is crucial. It drives catalytic activity and helps determine the molecule's final structure. Next, we explore how RNA is classified. There are 3 major classic types: mRNA, tRNA, and rRNA. We have coding RNA, which is mRNA. It carries the genetic code. Then we have non-coding RNA, which includes tRNA and rRNA. They are essential for protein synthesis, but don't carry the code themselves. And we have regulatory RNAs like miRNA and siRNA, which control gene expression. The slide includes a classification chart image. It's shaped like a central circle labeled RNA classification, branching out. One main branch leads to mRNA or messenger RNA. Another leads to tRNA, transfer RNA, and rRNA, ribosomal RNA. A third branch points to small RNA regulatory, which includes types like mRNA and siRNA. It's a great visual to see how these different RNAs are grouped by their function.
Let's start our detailed look with the messenger. This is messenger RNA, or mRNA. Its primary job is to carry genetic information from DNA in the nucleus out to the ribosomes in the cytoplasm. For protection and stability, a mature mRNA molecule has a 5' cap and a 3' poly-A tail. Its structure includes a coding sequence which is flanked by untranslated regions, or UTRs, on both ends. And ultimately, it serves as the direct template for protein synthesis during translation. The figure on this slide illustrates this journey perfectly. It shows a strand of mRNA represented as a line with dots for codons, traveling from a stylized nucleus on the left to a ribosome on the right. The ribosome is shown as a two-part structure reading the mRNA. This visual clearly shows mRNA as the information carrier between the genetic library and the protein factory. Moving forward, let's meet the adapter molecule, transfer RNA, or tRNA. tRNA is the essential adapter. It carries specific amino acids to the ribosomes during protein assembly. It has a distinctive clover-leaf structure when shown in 2D, which folds into a compact 3D shape. A key part of the structure is the anticodon loop. It's a relatively small molecule, about 76 to 90 nucleotides long, packed into that efficient 3D structure. Its genius is in recognition. It recognizes specific codons on the mRNA through perfect anticodon-codon pairing. The slide includes an image labeled Structure of tRNA. It shows the classic 2D cloverleaf diagram with its loops and stems clearly visible, giving us a sense of the complex yet orderly shape of this molecular translator. Now let's talk about the major component of the protein factory itself, ribosomal RNA, or rRNA. rRNA is the major structural and functional component of ribosomes, making up about 60 to 65% of their mass. Remarkably, it exhibits ribozyme activity, meaning the RNA itself catalyzes the formation of peptide bonds between amino acids. In eukaryotic cells, there are multiple types, like 18S, 28S, 5.8S, and 5S rRNA, each part of the large or small ribosomal subunit. It is absolutely essential for the protein synthesis machinery to work. The diagram on this slide shows the process of translation. We see a ribosome composed of a large and small subunit straddling an mRNA strand. A tRNA molecule is entering, carrying its amino acid. Arrows show the direction of translation as the protein chain grows. This image ties together mRNA, tRNA, and rRNA in their collaborative dance. Beyond the big three, there's a whole universe of other RNA types with specialized jobs. miRNA, or microRNA, is involved in controlling gene expression. siRNA, small interfering RNA, mediates gene silencing. snRNA, small nuclear RNA, is essential for the RNA splicing process. And snORNA, small nucleolar RNA, guides the modification of rRNA. The slide has a simple graphic listing these small RNA types, reminding us that RNA's roles extend far beyond just carrying a message. So, bringing it all together, what are the core RNA functions in gene expression? This slide shows the central dogma of molecular biology: DNA to transcription to RNA to translation to protein. RNA sits right in the center of this flow. To put it simply, RNA plays the central role in protein synthesis. mRNA carries the genetic information from DNA to the ribosomes. Through the processes of transcription and then translation, this information becomes functional proteins. And these proteins ultimately control all cellular activities and gene expression. It's the foundational process of life. But RNA's functions go even further into the realm of regulation. RNAs like miRNA and siRNA are key players in gene silencing. They control mRNA stability and degradation. They're involved in post-transcriptional regulation via splicing, editing the mRNA transcript. They even participate in chromatin remodeling and epigenetic control, influencing how DNA itself is packaged and read. This slide lists the sophisticated regulatory roles, showing RNA as a master conductor, not just a messenger. And one of the most exciting discoveries about RNA is its catalytic function. Ribosomes are RNA molecules that have catalytic activity. They can speed up biochemical reactions. Examples include self-splicing introns and the enzyme RNase P. Remember, the ribosome, the part that forms peptide bonds, is actually catalyzed by the rRNA, not a protein. This incredible fact supports the RNA world hypothesis, which suggests that RNA-based life forms could have been some of the earliest on Earth. RNA can both store information and perform work, a truly powerful combination. And that brings us to the end of our exploration. Thank you. We hope you enjoyed learning about RNA. Let's quickly recap. RNA is a single-stranded nucleic acid with vital, diverse functions. We covered the three main types, mRNA, tRNA, and rRNA, plus important regulatory RNAs. We saw its central role in gene expression and protein synthesis, and we discovered its amazing regulatory and catalytic functions that go beyond just protein coding. Thank you. See you in the next class.